Hello. Welcome back to the channel. I'm back with a new video, as usual. Before diving into the topic, hit the subscribe button and like and share the video. Today's topic will be about proteins. Let's begin by asking an important question. What are proteins and what role do they play in biological systems? Proteins are large, complex molecules that play many critical bodily roles. They are made up of amino acids, which are linked together in long chains. 20 different amino acids can be combined to make a protein. The sequence of amino acids determines each protein's unique structure and specific function. Understanding the structure and function of proteins depends on the amino acids that make up each specific protein. Let's explore what amino acids are and their characteristics. Amino acids are a group of organic compounds that contain two functional groups, an amino group, NH2, and a carboxyl group, COH. The amino group is basic, while the carboxyl group is acidic in nature. In total, there are about 300 amino acids found in nature, but only 20 of them are incorporated into proteins. Structure of amino acids. Each amino acid has a basic structure consisting of a central carbon atom, alpha carbon, an amino group, NH2, a carboxyl group, COH, a hydrogen atom, H, a distinctive side chain, R group that varies among different amino acids. Optical isomers of amino acids. Optical isomerism in amino acids refers to the existence of two mirror image forms, known as enantiomers, due to the presence of a chiral center. A chiral center is a carbon atom bonded to four different groups. In amino acids, the alpha carbon, the central carbon, is typically the chiral center, except for glycine, which has two hydrogen atoms attached to the alpha carbon and is therefore not chiral. Classification of amino acids. Amino acids classification based on structure. The 20 standard amino acids found in protein structure are divided into 7 distinct groups based on side chain properties. 1. Aliphatic amino acids. 2. Hydroxyl group-containing amino acids. 3. Sulfur-containing amino acids. 4. Acidic amino acids and their amides. Basic amino acids. 5. Aromatic amino acids. 7. Amino acid. Aliphatic amino acids have side chains that are non-aromatic and consist of either straight or branch carbon chains. These amino acids are generally hydrophobic because of their nonpolar side chains. Hydroxyl group-containing amino acids and their side chains are known for their polar and hydrophilic properties. Sulfur-containing amino acids have sulfur atoms in their side chains. The two main sulfur-containing amino acids are cysteine and methionine. Acidic amino acids have side chains that contain a carboxyl group, which gives them a negative charge at physiological pH. Basic amino acids have side chains that contain amino groups, which give them a positive charge at physiological pH. Aromatic amino acids are those that contain an aromatic ring in their side chains. Amino acids are a unique class of amino acids characterized by the presence of an imino group instead of the typical amino group. The most well-known amino acid is proline. Classification of amino acids based on nutritional requirements. Amino acids are classified into two major categories based on their nutritional requirements. Essential amino acids cannot be synthesized by the human body and must be obtained from the diet. Non-essential amino acids can be synthesized by the human body based on metabolic pathways. Glucogenic amino acids can be converted into glucose through gluconeogenesis. Ketogenic amino acids can be converted into ketone bodies. Both glucogenic and ketogenic can be converted into both glucose and ketone bodies. Amino acids as ampholytes. Amino acids possess both acidic and basic groups, allowing them to donate or accept protons. Because of this property, amino acids are classified as ampholytes. A zwitterion is a hybrid molecule that contains both positive and negative ionic groups. In aqueous solutions, amino acids primarily exist as zwitterions, in which the amino group is protonated, NH3+, and the carboxyl group is deprotonated, COO. This form is electrically neutral, but contains both positive and negative charges. Amino acids rarely exist in a neutral form with free carboxylic and free amino groups. Peptides and proteins. Peptide bonds are the chemical bonds that link amino acids together to form proteins. They are formed through a dehydration synthesis reaction, where the carboxyl group of one amino acid reacts with the amino group of another, releasing a molecule of water, H2O, and forming a covalent bond. Structure of proteins. The sequence of a protein is determined by the DNA of the gene that encodes the protein or that encodes a portion of the protein for multi-subunit proteins. A change in the gene's DNA sequence may lead to a change in the amino acid sequence of the protein. Even changing just one amino acid in a protein sequence can affect the protein's overall structure and function. To understand how a protein gets its final shape or conformation, we need to understand the 4 levels of protein structure: primary, secondary, tertiary, and quaternary. Let's begin our discussion on primary protein structure. Before we dive into that, it's important to understand a key point regarding protein structure. The 4 levels of protein structure differ from one another based on the types of bonds or interactions involved, the groups that facilitate these interactions, and the number of polypeptide chains present. Primary protein structure refers to the linear sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code and is held together by peptide bonds. The primary structure is crucial because it dictates the protein's final 3-dimensional shape and function. The secondary structure of a protein refers to the local folding of the polypeptide chain into specific patterns, stabilized by hydrogen bonds. The 2 most common types of secondary structures are alpha helix and beta-pleated sheet. In secondary protein structure, the R groups do not participate in interactions. The tertiary structure of a protein refers to its overall 3-dimensional shape, formed by the folding and interactions of its secondary structural elements, alpha helices and beta sheets. This structure is stabilized by various interactions among the amino acid side chains, including hydrophobic interactions. Nonpolar side chains tend to cluster together in the interior of the protein, away from water. Hydrogen bonds form between polar side chains and contribute to the stability of the protein structure. Ionic bonds occur between positively and negatively charged side chains. Disulfide bonds, covalent bonds between the sulfur atoms of cysteine residues, providing additional stability. The quaternary structure of a protein refers to the arrangement and interaction of multiple polypeptide chains, subunits, in a multi-subunit complex. Remember, protein structures from primary to tertiary consist of only one polypeptide chain, unlike quaternary protein structures which involve multiple chains. The last topic in protein part is protein denaturation. Protein denaturation is the process by which a protein loses its native 3-dimensional structure due to the disruption of non-covalent interactions and disulfide bonds. This structural change results in the loss of the protein's biological function. This is all for today's video. I will come back soon with a new topic. Until then, I wish you all peace.